Oh, good morning, dear friends. Thank you very much for watching DrugTimes Roadshow. This is our second session, co-organized by DrugTimes and UCAM. This is my background. Okay. So first of all, please kindly allow me to present a quick background introduction. DrugTimes is a new media-based platform. We are proudly to have over 200,000 professionals in our network. Our articles and videos have been watched for over 22 million times last year. And over the past few years, we have contributed to the successful completion of several BD deals, including cross-border transactions. The total value is about $300 million. Discovery and development of ADC drugs has been very popular and hot over the past years. And this area is also, frankly speaking, rather crowded. Days ago, Pfizer announced the agreement to acquire Seagen, an ADC drug development veteran, for $43 billion. Within 1 year, MSD, another big MNC companies, well-known MNC companies, has completed 3 deals with Kelun Biotech in China for non-drug ADC drugs. Total value is about $10 billion. In 2023, this year, over the past maybe 70 days, and China pharma and biotech companies have successfully completed 10 license out deals to Western pharmaceutical companies. Total value is over $7 billion, totally 10. Among these 10 deals, 4 of them are ADC related. The total value of ADC related deals is about $3 billion. As you can see, this area is very, very active. So from the China pharma and biotech perspective, the latest ADC deal is shown on this slide. It's between KMed Biosciences, Lepu Biopharma jointly with AstraZeneca, another MNC, big company, big MNC company. The upfront is $63 million and the milestone $1.1 billion. We at DrugTimes, like radar, we are screening, we are monitoring the industry every day, including the ADC related good news progresses. And we also organize or co-organize several events on the ADC, and including the news, the DS8201, currently the most famous ADC drug. It was approved in China, not on February 24th. DrugTimes is the first new media in China to share this news. And so far, this article is the most read one. It's close to 100,000 professionals, friends have viewed our article. So today, we are much pleased and honored to invite AbTis, a leading ADC biotech in Korea, to present. First of all, I will ask co-host today, Dr. David Kwan, CEO of UCAM, to introduce himself, and Professor Jiang, CEO of AbTis. After presentation by Professor Jiang for about 40 to 45 minutes, we will have a Q&A session. Mr. Richard Yang, DrugTimes BD. Okay, please. So I'm back, right?
Yeah, just.
Oh, sorry. Yeah, sorry
perfectly.
for the internet connection problem. So
Okay.
basically, I will invite Dr. David Kwan, co-host of today's webinar, to introduce himself, and then introduce Professor Jiang, CEO of AbTis, today's speaker. Okay, David, please. Thank you.
Okay, thank you, Dr. Guo. Hello, everyone. My name is David Kwon, and then I am the manager of the UCAM in Korea. UCAM provides CRO and CDMO consulting service for the pharma and biotech industry in Korea. As a member of the Global Pharmaceutical Key Opinion Leader in KHID, I do CRO and CDMO committee member and try to support new drug development and the licensing deal. I want to express my gratitude to Dr. Guo and DrugTimes for giving me the opportunity to introduce one of the excellent ADC pipeline from Korea. Today, I'm thrilled to present to the AbTis, a leading ADC new drug biotech company in Korea, and its CEO, Professor Jung Sang-jeon, who is also the Dean of the School of Pharmacy at Seungkyunkwan University. Through this presentation, I hope to provide you with insight into AbTis' innovative ADC platform and technology for developing drug candidates. My ultimate goal is to foster collaboration and the licensing deal that will benefit both our industry and patients. Furthermore, I hope that this presentation will force opportunity for collaboration and the licensing deals in the near future. As we continue to showcase Korea's new drug pipeline, I'm excited about the prospect of sharing more of our country's innovative drug candidates with all of you. Thank you and enjoy Dr. Jung's presentation. Dr. Jung, please.
Okay, thank you, Dr. Kwon. Before Professor Jung's presentation, I would like to apologize quickly because we are using Zoom, which is far, far away. And also today, Shanghai it was raining heavily. So sorry for the technical issue. Yeah,
okay.
I'm sure you will enjoy Professor Jiang's excellent presentation in the coming about 45 minutes. Okay, Professor Jiang, it's all yours. Thank you.
Yeah, thank you. Thank you, Dr. Kwon and Dr. Guo. So for making this event, wonderful event. So hello, everyone. This is Sang-jeon Chung, the CEO of AbTis, which is a leading ADC biotech company with the world's best platform ADC linker technology. Okay, this slide shows executive summary. So AbTis has developed third generation ADC linker platform, so-called AbClick. AbClick can give site-selective conjugation to unmodified antibody. And then AbClick can control the PK of ADC product by choosing AbClick Standard or AbClick Pro linkers. Currently, AbTis has developed new pipeline, Claudin 18.2 targeting ADC. Then we aim to do IND filing in first quarter of the next year. So this Claudin 18.2 is highly attractive target, thereby AstraZeneca, BMS, and MSD entered anti-Claudin 18.2 ADC competitions. We expect we can make over $1 billion of licensing deal. And then currently, AbTis is focusing on ADC pipeline development. Then AbTis platform can be extended to antibody radionuclide conjugate and antibody peptide or protein conjugate or antibody oligonucleotide conjugate. Using this technology, we can also develop companion diagnostic reagent. Then currently, AbTis is undervaluated because of rising interest rate and weak investment sentiment in Korea. So we expect we will have very high upside potential. Current valuation is $50 million. So also because we have a very specific and excellent ADC linker technology, probably global biopharma will be interested in the M&A activity. Okay, AbTis was founded in year 2016, and then we have got funded Series A and Series B until year 2021. And currently, we are preparing for GMP production and GLP talks. So we are doing the Series B bridge funding. That's why I am presenting here. So until now, leading venture capitals and asset management companies in Korea have invested. So there are, this slide shows, the venture capital companies and management companies. We have very excellent executive management teams. I, by myself, measured organic chemistry in PhD course, and then did research work in biological science at Harvard University. Then currently, I am a professor of School of Pharmacy, Sungkyunkwan University. Then I have been involved in many drug discovery activities. And then Dr. Choi Won is a MD, PhD. He had worked at Merck Korea and GSK Korea, LG Life Science, and Iltong Pharmaceutical Company. Then he had many experience in drug development works. And then Mr. Jung-Jin Kim, he is a pharmacist, and then he worked at Dream Pharma and the JL Pharmaceutical Company as executive vice president in plant management. Also, we have very excellent scientific advisory board members who are working in toxicology area and animal models, and also clinicians in Seoul National Hospitals. Dr. Kang is worked in the nuclear medicine area, and Dr. Jung is expert in the antibody development. And Dr. Jeo Jung is a surgeon, and he is expert. He has expertise in gastrointestinal surgery. So currently, we are developing ADC pipeline for gastric cancers. He is helping us in many ways. Okay, linker is very important in ADC development. So ADC linker can be divided into two groups. First one is conjugation linker, antibody, and payload releasing linker, which determine where and when payload is detached from the antibodies. So this conjugation linker includes three generations. First one is random conjugation linker, and second one is site-specific conjugation linker. This site-specific conjugation linker includes site-directed mutagenesis of antibody, and then the mutated amino acid has been used for drug conjugation. The third generation linker is a site-selective chemical conjugation linkers, which AbTis has developed. So this site-selective chemical conjugation linker does not require mutation of antibody. It can directly conjugate the drug onto a specific amino acid of the antibodies. And then payload releasing linkers include enzyme-mediated cleavable linkers. This is the representative cleavable linkers. Then these linkers can be cleaved by cadepsin or glycosidase or sulfatase, phosphatase, etc. These enzymes are overexpressed in tumor cells. So there is huge difficulty to site-selective antibody conjugation because antibody is a large molecule consisting of over 1,300 amino acids. Usually lysine and cysteine residue have been used for drug conjugations. One antibody includes around 90 lysine residues. So it is almost impossible to selectively modify one or two of lysines from these 90 lysines. And also, many companies are using cysteine for drug conjugation, but also cysteine also is included in the antibody. Around 32 cysteines exist. So by partial reduction of this disulfide bond, drug is attached. So here, I would like to give you information about drug conjugation method and related to drug stability. So when you conjugate drug using lysine, there are amide bond is formed. This amide bond is quite stable in the plasma. However, if you use cysteine and maleimide conjugation method, the thus prepared ADC is easily oxidized in the blood vessel and then undergo the elimination reaction result in free drug causing the cytotoxic side effect. So if possible, you better choose lysine conjugation for ADC development. Okay, there have been developed 3 generations of ADC linkers. The first generation ADC conjugation linker is a random conjugation linker, which results in a mixture of ADC product. So during the large production, you have to purify these portions to obtain drug substance. So it makes CMC very difficult. Therefore, many companies have developed second generation ADC linkers. This second generation ADC linkers introduce site-directed mutagenesis of the antibody. So this mutation should be obtained in the plasmid, the DNA level. So DNA which encode antibody should be mutated and then transport the cell line and has to be prepared in stable cell line for large production of antibodies. And then this mutated amino acid can be modified with drugs. So this method gives homogeneous ADC product. Let's compare the characteristics of first and second generation of ADC linkers. The first generation ADC linkers cannot control drug antibody ratio. However, this method can use original antibody or biosimilar antibodies without any modifications. And whereas second generation ADC linkers can control drug antibody ratio and the site of drug conjugation. However, this method cannot use original antibody or biosimilar. So to use this second generation ADC linkers, you have to produce new antibodies. So the third generation ADC linkers has advantage of both methods. So this third generation ADC linker directly introduce the drug onto the specific amino acid of the antibody. So it can control the drug antibody ratio, and also it can use original therapeutic antibody or biosimilars. Okay, we have developed site-selective chemical conjugation linkers. The principle of our site-selective antibody conjugation linker is described in this slide. So this is AbClick. This AbClick is a kind of peptide decorated with specific chemical functional groups. This AbClick tightly binds to the cleft between this CH2 and CH3 domain of antibodies. And then this functional group located in a very close to the lysine 248. So this lysine 248 can attack this functional group to form a covalent bond. This chemical reaction occurs only when this AbClick binds to the antibody. However, if you incubate this linker with lysine amino acid in the physiological buffer, there is no reaction proceed. So which means this AbClick linker gives a very specific conjugation to lysine 248 of antibodies. So core competence of AbClick can be explained by this AbClick can give site-selective conjugation to unmodified antibody with a defined DAR, resulting in a metabolically stable isopeptide bond to only lysine 248. Then our AbClick can control the PK property by choosing a subtype of AbClick linkers. Also, this linker technology can employ diversity of payload. So basically, any type of payload can be introduced to the antibody by using our linker technology. Thus prepared ADC product is very stable in serum. However, it is easily cleaved in tumor cells. So currently, we are using two subtypes of AbClick linkers. The first one is AbClick standard, which introduces drug through FC binding peptide. This FC binding peptide decreases the biological half-life of antibody conjugate. Therefore, this linker can be used for companion diagnostic agent or radioimmunotherapeutic drugs. And then AbClick Pro directly connects drug onto the lysine 248 without this peptide. And thus prepared ADC product has a longer biological half-life, very similar to the other ADC product. Therefore, this linker technology has been used for ADC preparation to treat solid cancers. So the last year, end of last year, we made ADC partnership agreement with Lonza. So Lonza, as you know, Lonza is the #1 CDMO company in the world. So they are producing drugs, small molecule drugs, and also antibody and protein drugs. Recently, they are preparing ADC toolbox, which means any drug developer asks Lonza. Lonza can produce small molecule payload and antibody, and then conjugate small molecule antibody using ADC linkers. So Lonza chose our technology for building their ADC toolbox. Through this partnership agreement, AbTis can promote our AbClick technology to global big pharma. And also, we can expand the partnership with many ADC biotech in the world. Then we expect a continuous revenue from upfront and milestones. Okay, AbClick can be extended to many different types of drugs. Currently, we are producing antibody-drug conjugate for anti-cancers. And also, we are testing antibody radionuclide conjugate for companion diagnostic and radioimmunotherapies. Also, we can expand our technology for antibody peptide or protein conjugate for long-acting biopharmaceuticals. And also, we are planning to develop antibody oligonucleotide conjugate for drugs with totally different mode of actions. And we have constructed pipelines, several pipelines, and then Claudin 18.2 is a leading pipeline, which we are aiming for GLP tox study in this year. So the key R&D data of Claudin 18.2 targeting ADC is summarized here. So we obtained the efficacy, the good efficacy, with the 2 milligram per kg of MED values. This represents the efficacy of our ADC against animal models. And also, we obtained 10 milligram per kg of MTD, which indicates the toxicity. And therefore, we obtained 20 of therapeutic index. This is the highest value among the ADCs developed until now. Also in CMC, the titer of antibody is up to 6.4 gram per liter. The final ADC yield from the antibody is up to 65%. This is also very high valued. So we are aiming to identifying the first quarter of the next year. And then also we apply for clinical trial, phase 1 clinical trial in the second quarter of year 2024. This will give our ADC pipeline fourth or fifth clinical trials in United States. The indication is aimed for gastric cancer and pancreatic cancer and lung cancers. We're planning to do the clinical trial in orphan drug designation or breakthrough therapy designation for quick approval from FDA. So let me introduce our leading pipeline of Claudin 18.2 targeting ADC. The market size of gastric cancer is expected to 7.1 billion by year 2026. Unfortunately, there is no targeted anti-cancer for gastric cancers. Herceptin-based drug is only targeted anti-cancer for gastric cancers. However, only 20% of gastric cancer patients are overexpressing HER2. Therefore, the other 80% of patients cannot be treated with Herceptin-based drugs. Interestingly, around 87% of gastric cancer patients overexpresses the Claudin 18.2. Thereby, many big pharmaceutical companies are interested to develop ADC, which target this Claudin 18.2. So we prepared ADC, so-called the code name AT211, using anti-Claudin 18.2 antibodies. Our ADC AT211 includes only 2 drugs per 1 antibody. And also we prepared AT208, a zolbetuximab MMAE conjugate with DAR 4.6. So we compared anti-cancer activity of AT211 with this AT208 and naked MMAE drugs. Very surprisingly, our AT211 showed strong anti-cancer activity in Claudin 18.2 overexpressed cell line, such as MIA-PACA2, antigen overexpressed, and SNU601, which are natural cancer cells. So as you can see here, our ADC showed very similar anti-cancer activity with even naked drugs. However, our competitor showed very poor anti-cancer activity against MIA-PACA2 and SNU601. Also, this drug shows cytotoxicity against marker-free cell lines, which means it can cause a side effect. So, using this gastric cancer cell SNU601, we prepared the CDX model and tested for, and then AT211 was tested for anti-cancer effect against this CDX model. By single injection of 2 or 4 MPK of AT211, tumor tissue shows total complete regression. However, AT208, our competing drug, did not show significant anti-cancer activities. This is the tumor tissue after sacrificing the tested animals. So from this study, we determined the MED value of AT211 to be less than 2 MPK. And currently, we are doing this efficacy test using more fine dosage of drugs from 1 MPK to 2 MPKs. We expect to obtain around 1.5 MPK of MED from the current animal study. So we studied the stability of ADC and compared with that of zolbetuximab MMAE conjugate. At the beginning, I explained you lysine conjugation of drug is very stable. However, cysteine-mediated drug conjugation gives poor stability. So we observed the same result. Our ADC showed no significant cleavage of drug from the antibody. However, our competing drug shows significant cleavage of drugs. So this line, a faint orange line, represents the total antibody, include drug-attached antibody and drug-free antibodies. This deep orange cut lines represent only drug-attached antibodies. So you can see around 20% of difference between free antibody and free drugs. However, in our ADC shows very similar amount of free antibody and total ADC. So it also, our drug was tested in vivo pharmacokinetic studies. Here, free antibody and total ADC shows almost the same amount, which means our ADC is not cleaved in vivo. So the biological half-life was determined to be 4 to 5 days with our 8211. Then we have finished the pharmacological study and non-clinical study. We finished pharmacokinetics using rat model. Also, we finished non-GLP monkey toxicity. And then we are planning to do GLP tox using monkey and rat. So currently, our ADC has been produced by WuXi AbTis in China side. Then they finished the condition for antibody production and link and payload and bioconjugation productions. So we are planning to do GLP tox study in the second quarter of this year. And then we are filing for IND. We will submit for IND approval. So this is, we have tested for non-GLP monkey tox. So in the first stage, we dosed it for 8 and 12 milligram per kg of 8211 for the monkey. But the first trial, one monkey in 12 mpk dosage animals, one monkey died. So we decreased the dosage to 4, 6, and 10 milligram per kg. And all the animal in these conditions, all tested animal were well tolerated. So our CRO cannot figure out the causal relationship between the test article and the death. So death is obscure. They said there's no relationship between the death and drug toxicity. So they suspect the condition, health condition of the tested monkey was not so good. So anyway, we obtained more than 2 mpk of MED and 10 mpk of MTD. Therefore, therapeutic index was estimated to be over 20. This is the highest therapeutic index among the competitors. So we compared the anti-cancer activity and toxicity with those of our competing drugs. So this LM302 has been transported from LaNova to Turning Point. The deal size was $1.2 billion. Then they disclosed MED value of 10 mpk, even though they are using same payload with our 8211. So our ADC include only 2 drugs for 1 antibody, but our competitors have more than 2 drugs. So LM302 did not disclose the DAR, but we guess they have more than 2 of DARs. So this SOT-102 are using highly toxic payload. So their MED value was announced to be 0.2 mpk, and HNSTD was to be 0.6 mpk. So their therapeutic index is around 12.5, but still much lower than our ADC. So I heard recently NO2 was approved from Chinese FDA for drugs. So we compared our 8211 with Kadcyla NO2, which prepared from herceptin. Then it can be used for gastric cancer treatment. However, as I told you, only 20% of gastric cancer patients are expressing this the antigen, so it cannot be used for majority of gastric cancer patients. So let's see their therapeutic index. In case of Kadcyla, therapeutic index is less than 6, and therapeutic index of NO2 is less than 12. However, 8211 shows more than 20 of therapeutic index, which implies our ADC pipeline is very safe anti-cancer drugs. So we have funded twice, and then currently we are getting funding a Series B bridge for GMP production and GLP tox. Our value is listed here. So after this Series B bridge funding, we are planning to minimize the investment by making money through licensing out upfront and milestone fee for R&D fund. So our vision is we want to be a global top 3 ADC company. So we aim to develop at least 5 new ADC pipelines by year 2030. So we are sure it will make AbTis to be a global top 3 ADC company. So here I want to propose several types of collaborations. So we can license out Claudin 18.2 ADC pipeline if there are any companies interested to develop this pipeline. Also, AbTis is responsible for FDA IND submission, and licensee proceed from clinical trial phase 1. Or most favored model for this licensing deal is to develop our ADC pipeline together, and then just to put the same development budget and then share the profit. Now also, we want to receive strategic investment or financial investment from the Chinese side. And then also, if the Chinese side company just give a strategic investment, AbTis will give our AbTis asset license first. Then also, there is M&A opportunity for continuous drug development, because currently Korean investment market is almost frozen, but we want to continue this drug development process. So if any company who has enough development budget want M&A, and then we will agree with the company. And also with the M&A, even after the M&A works, we will give active technical support. And also, we think this is a really good opportunity to invest, because AbTis is currently undervalued because of Korean investment market is poor. Okay, this is all I have for today's presentation. So I'll be happy to get the questions if you have. Thank you.
Okay, thank you very much, Professor Jiang, for your presentation. Very complete, detailed, and thorough. And also your technology and the current pipeline are very impressive. Thank you. Okay, now let's move on to the Q&A session. And Dr. David Kwan and my colleague, Mr. Richard Yang, we will go online too. And 4 of us will answer any potential questions from the audience, including technical questions and BD-related questions. Okay. Actually, this is our second roadshow. It got a lot of attention even before our live broadcast today. Some Chinese biotech companies already show, include also big farmers in China, also already show the interest in our AbTis. So we even received the questions from our friends before today's roadshow. So I will start with I will read the questions and then ask Professor Jiang to answer. Let's start with a simple one. So, Professor Jiang, your company's name is AbTis. Could you please explain the meaning so that to help us remember the name of your company?
Yeah.
It's a simple one.
Okay. When I named my company, I had spent a lot of time, and then finally I decided AbTis, because Ab means antibody and Tis means technology. So the meaning of AbTis is antibody technology. It's very simple and robust meaning.
Okay, thank you.
Okay.
So Professor Jiang, maybe could you please turn off your slideshow so that we can our friends can see all of us on the screen? Yeah.
Yeah, slide is here. So
yeah, we can turn it off for now. If we need, we can reload. Okay. Good. Yeah.
Okay, it's okay.
I mean,
Professor,
turn off your
slideshow.
Right.
Okay, hold on.
Yeah, take your time, please.
Okay.
Good. OK. Max 我们4个人都上镜啊。No. Okay. The next question is a technical question, Professor Jiang. So, as we can see, from your presentation, from your PPT, you have different DAR value. I mean, we know the DAR varies, right? Could be 2, could be 4, 6, or HER2 almost 8, right? 7.7. So could you please kindly comment on the DAR value, whether it is the higher the better, or it doesn't have to be like that? Yeah, thank you.
Okay, that's a good question. So everyone knows, anti-cancer drug is also toxin. So you want to have just the minimum amount of toxin, but you want to treat your cancer with minimum amount of toxin. So in my personal opinion, if you can use lower DAR and then cure your cancers, you better use lower DAR ADC. And also, if you increase the number of drug per antibody, the corresponding ADC has lower water solubility, which causing the low biological half-life. So you better use low number of DAR. But you better maybe you may ask, how about the NO2? NO2 has DAR 8. So in that case, I read some paper from Daiichi Sankyo. They compared DAR 4 ADC and DAR 8 ADC for pharmacological efficiency. So their paper showed DAR 4 and DAR 8 ADC showed very similar final pharmacological activities. But during the development, they chose DAR 8. I guess that's because DAR 8 is more easy to establish CMC compared to DAR 4 or DAR 2. So that's the biggest reason why the Daiichi Sankyo chose, DAR 8 for NO2. It's my personal opinion.
Okay, thank you.
Yeah.
Okay, let's move on to a third question. The third question is about your product AT211. As you know, your AT211 is targeting Claudin 18.2. Claudin 18.2 is a very, very hot, right? A hot target.
Yes.
Actually, a few years back, during the ASCO, the Claudin 18.2, I mean, was an underdog. Oh, it got attention worldwide. So, as we all know, that for gastric cancer, which is very common in China, in Asian countries, right? It's a much better target than HER2. So, but this area is so crowded. What is the advantage of your product in development, AT211? Could you please comment? Thank you.
As I showed the comparison slide in my presentations, most ADC pipeline to target Claudin 18.2 are in clinical trial phase 1. So we are in the preclinical stage. When we compare our AT211 with other competing pipelines, AT211 shows definitely much safe toxicological properties. So we believe AT211 has much more advantage over other competing pipelines. So we want to explain that fact to our partners. Then I'm sure we can catch up, the other competing ADCs to target Claudin 18.2. That's why, I do not stop the development even in this, difficult situations. So that's why I want to get fund and I want to move forward for clinical trials. So I need good friends and good collaborators to make it successful.
Okay, thank you. So we will work with Dr. David Kwan to help you find a quality partner. Okay, let's move on to more questions. Okay, David, please prepare for one question for you soon. After two more questions. Okay. The second question is, as we know, to develop a good drug, the drug has to be safe, has good efficacy, and at the same time, the cost is also a key factor, right? So, yes. Professor Jiang, could you please comment on your on your platform, AbClick? Does it have any CMC cost advantage?
Yeah, we are using peptide linkers. Every site-selective linker use, reactive functional group with the cysteine or the lysine residue. So everyone has to use that function. And also, most ADC include cleavable linker site. So the component for ADC preparation is very similar with other ADC. But our technology does not require the site-directed mutagenesis of antibody, and we do not need to produce new antibodies. So considering that fact, our linker technology has a great benefit, great advantage in the preparing cost compared to other ADC technologies.
Okay. So when it comes to the therapeutic window, therapeutic index, it's also AT211 has about 20, right? What
Yes.
about other ADC products produced by AbClick technology?
Yeah, I believe.
Case by case,
yeah,
right?
Sure. Yeah, right. Definitely we can improve the therapeutic index because NO2 and Kadcyla include maleimide and cysteine conjugation unit. So I believe that causes the toxicity because it is cleaved in the blood vessel of our body. So if you replace the cytoether bond with the isopeptide bond, definitely the resulting drug will increase the therapeutic index.
Okay, thank you.
Yeah, actually,
okay, another technical.
Okay.
Yeah. Okay, another technical question for Professor Jiang is a friend is watching our show, Roadshow, and he Tao, is Tao from China. Our friend Tao, he missed the beginning part. So his question is, what is the payload of your ADC? And maybe, as an extended question, could you please comment on other payloads? Have you considered other payloads as well? Yes, please.
Sure, yeah.
Right.
Actually, we have tested MMAE and then SN38, and then now we are preparing exatecan as a payload. So basically, and also we are preparing DNA alkylating agent, duocarmycin. So we are preparing several, potential payloads. Then one of the most exciting parts should be when we make ADC with exatecan and then compare with NO2. So what happened between our ADC pipeline and NO2? That will be very interesting. So basically we can
thank you.
type of payload. Yeah.
Okay, thank you. Professor Jiang, please have some water or tea or coffee. Okay. Have a quick break.
I need that.
Yeah. The next question is for Dr. David Kwan. It's a general question. Actually, over the past years, more and more collaborations between companies in both countries, right? Now, friends, professionals in China are curious. Today, how many biotech drug development companies in Korea these days? About
the biotech companies, they are listed around over 400 biotech companies. Not only small molecule, but also biologics, and some are diagnosis area. So yeah, over 400 biotech company, they are study on their new drugs.
Oh, 400. Oh, okay, thank you. Okay, the next related question is, when a biotech company in Korea is ready for IPO, will they choose Kosdaq or Nasdaq or other public markets?
Yeah, depends on the project. Some are most of the biotech in Korea, they are Kosdaq market. They are focusing on the Kosdaq market at first. And then few of them try to in the Nasdaq also, but no Chasdaq.
Oh no, okay. Okay, thank you. Thank you, David. So please have a rest. And if more questions, I will ask you to help. Okay.
Okay,
so,
thank you.
A related question. This one maybe for Professor Jiang. So what is your plan for IPO? So you will go to Nasdaq? It's a related question.
Actually, we are aiming to Kosdaq, IPO to Kosdaq.
Okay.
Actually,
Okay, thank you. The answer is very clear. Yeah. Okay, so you mentioned the collaboration between AbTis and Lonza. It's a very, very good one. Congratulations! It was last November 24th. So this is non-exclusive, which means other companies can form a similar collaboration with AbTis too, right?
Yeah, this is the actually exclusive condition. So, but it's for only CDMO companies for CDMO purpose. But any company can collaborate with us, and any company can do the direct licensing deal for using our linker technology. And then the company can choose any CDMO company. It doesn't matter.
Okay, thank you. Okay, the next question is also BD related about So, for a potential collaboration between AbTis and let's say a company in China, biotech or pharma company, so the collaboration could be either based on a specific product like AT211, and in the meantime, it could be based on your technology, right?
Yes. Or
you are open.
We are open for any type of collaboration. If actually, currently we are discussing with one Chinese company for co-development, and the company has their own antibody and their own payload, so we can help them.
Right. Yeah. Okay, good. You are also open to M&A possibility, right?
Yes.
Okay, that's good to know. That's good to know. Okay.
Because drug development require lots of big size of budget, so if any company who has a good financial conditions, it will be a really great chance.
That's right. So another question from our friends is about the patent. So how long is the patent? Life right now?
Yeah, actually we have several patents, and the key patent we have applied for the patent. So several companies are for evaluating our article. So we are just continuously apply many the series of the patents. So we are waiting for the evaluation.
Okay. Yeah, I see. Okay, good. So Dr. Kwan, do you have any more addition or comments?
I think this is the first chance to introduce the Korean ADC pipeline. And also in Korea, many of the biotech or some pharma company, they are also interested about the collaboration with the China and US partners. So if you can give some more chance, I would like to bring more pipeline to DrugTimes and then try to have more chance. Thank you so much for your support.
Okay, thank you, Dr. David Kwan. Oh, Professor Jiang, right now we have almost over 50 professionals are watching. I believe we will have even more. And this one can be replayed and reviewed anytime. And from today on, do you have anything, any thoughts you would like to share with the audience watching our show?
Mm-hmm. Yeah, just actually, I wanna keep developing our pipeline. So I want to find a good collaborator from your side.
Right.
That's what I want. And then
okay, sure. Actually, we have one more question. So, what is your business model or your strategy? So, you would like to develop AbTis into biopharma, later a pharma, later a MNC, or you would like to stay like a biotech as a discovery engine?
Okay, so basically, we have two types of business model. One is supply the ADC linker platform technology for developers. Also, we want to develop our own pipeline, and then we want to move for drug development company. So, yeah, it's
okay. Okay, good. Yeah, excellent. So yeah, it's almost 1 hour. It's almost 1 hour, and a lot of information. And I think your technology has advantages, and we do believe more and more friends and companies, biotech companies and pharma companies in China will be interested in and will work closely with Dr. David Kwan on promotion. And Dr. David Kwan, please be ready to be maybe very busy in the coming days.
I hope so. I would like to be busy, so don't worry about
good
my
busy.
time.
Busy.
Yeah, good. Yeah, I will do my best for the collaboration. Thank you.
Excellent. Actually, Mr. Richard Yang and the BD head of DrugTimes is also online. So, Richard, do you have any questions or comments?
I don't have any comments. If our audience have interest, please contact me as soon as possible. Thank you.
Okay. Actually, and Richard is working very diligently, and right now 40 projects are under his management, and AbTis is one of them, and we will move on very quickly, very efficiently, and hopefully we will be effectively. We will get a good result, and in the end, we will work together to develop good and safe and cost-effective drug for patients in Korea, in China, worldwide, right? So this is our common goal. We have the same end point, right? We have the same end point.
Exactly. Yeah, right.
Yeah. Okay. Questions or comments, maybe we can conclude today. And Professor Jiang has shared a lot of information, and we look forward even more good news from your end. And then we will work continuously with Dr. David Kwan and his company, his team, to first of all help Professor Jiang find quality partners in China and beyond, and then we will have more transporter collaborations. Yes,
right.
Yeah,
good.
Okay. Thank you, Dr. Guo.
Yeah. Thank you very much. Have a nice day. Yeah, it's lunch time, right? Okay. Many thanks to audience watching us today's show. Yeah, and have a good day. We will keep in touch. More coming to us. Yeah. Thank you. Bye.
Okay. Bye.
Thank you, David. Bye. Thank you. Yeah. Bye.